Hi, and welcome to this next tutorial on how to run statistical tests and generate graphs using the PECAn software. In this example, I'm actually going to launch it from using run it locally using RStudio. So what you do is just open up the server in the UI file and click this button here in the top right corner. It says Run App, and it's just going to launch it, and it's going to work pretty much exactly the same as if you used a web browser. So let's do something a bit fancy. Let's look at how do we do sort of multivariate tests and sort of regression analysis. So first thing we got to do is upload our whole disk analysis again. And assign our experimental conditions as normal. Then we're going to come up to see here where it says specify analysis type. If we leave it at default, it's just going to be the analysis that we ran in the sort of last tutorial. But let's do a complete single cell analysis. And now it's asking us to add an extra file. And this is where we add in the single cell tracking analysis files. So these guys are a bit of a special case. You can only add in these guys if you've already added in the whole disk analysis files. Basically, these are really big files, so it just saves memory on your computer to do it this way. Press open, and when that's done, press analyze. And if we looked at the analyze data table, we would see that we've got a lot more rows, and that's because each row is not a sort of single sample. Each row is a single cell. And so we've got the sort of the individual properties, sort of how far it is from the center of the ROI, its distance from sort of the clone perimeter, but also we've got the population level sort of measurements there as well, such as just sort of the overall clone volume and the overall percent clone coverage of the pouch. So now all that information is there. So let's go to the regression analysis tab, and let's choose sort of our regression type. So we have several different options. You just have to choose whichever one is sort of appropriate to your purposes. So let's start using a logistic example. And logistic regression is sort of for where your dependent variable is binary. So it's either going to be a yes or a no, or a 1 or a 0. So let's look at whether or not our cell is dying or not. So is it DCP1 positive? This vocabulary here is specific to sort of this experiment. Or it's specific to sort of the competition vocabulary and the way that I chose to label things. It'll look a bit different for you. And then we can choose our different options, sort of different predictor variables which might influence this pathway, but let's just kind of take these sort of defaults. So just like how big is the pouch, what's the loser clone coverage, and sort of what's the genotype, or what's the condition. And then when you do this analysis, you're making lots of sort of pairwise comparisons. So you want to do some sort of P adjustment if you're doing a decent number of variables. And we're also going to make a correlation matrix. And you can choose which test is most appropriate sort of for your data. Now we come up here and it's run that regression. So I'm not going to do a sort of whole tutorial on, how to evaluate and interpret logistic regression results. There's lots of resources out there, books and YouTube videos that can give you a good sense of how this stuff works. But the basics are down here. We've sort of got our numeric outputs with our p-values, with the p adjustment, and sort of our odds ratio effect sizes with our confidence intervals. We've also generated these sort of charts here. So what you're looking at for these charts is, this is to kind of give you sort of a intuitive sense of what the model that your regression has made is doing. So the way these work is the regression model is going to take in each of your sort of factors into account. But here we're just looking at 2 of them, sort of your dependent variable, which is this one, and a given predictor variable. For all the other variables, it's just putting in sort of the mean value. And then for like a typical cell, how does changing the ROI volume, increasing or decreasing it, influence the probability that a cell is dying in this case. and we can choose to display multiple of these ones or not. And once you've done that, you can sort of download your plot output as before. But when you're doing sort of these analyses, they are a bit more complicated than doing like a t-test or Wilcox. So, there are assumptions that are being made, and you sort of have to evaluate whether or not your model is appropriate. So what's going to be put out in the assumptions testing is going to depend on what sort of regression you do. Logistic regression is a bit more permissive than the other ones. So one of the major things we want to make sure is that our predictor variables aren't sort of auto-correlating. And so a test for that is the variance inflation factor. So we've got that there. We also have a correlation matrix, which is a fairly complicated plot. So it's going to take a bit of time for that to actually load, but it will do that. And I'll come back to it. Oh, there it goes. So what you're looking at here is sort of your correlation score and the significance of it, of your variables here. So for instance, this is the dependent variable versus the sort of genotype, and the dependent variable versus the size of the ROI, or the dependent variable here versus the percent clone coverage. It's just showing you sort of what sort of levels of correlation you're seeing. And we also have produced a number of sort of diagnostic plots to kind of help you get a sense of what's going on. So here is the observed probability in your data set versus what your model is predicting. And here's some sort of classic sort of diagnostic plots as well. So you can kind of look for things that could be messing with your analysis or whether it's valid or not. And if you work with R before, you might be sort of more comfortable or more familiar looking at sort of these kind of outputs. And this is just sort of the raw R output. And also here is our pseudo R squared calculation. And if we just kind of take a look at the pseudo R squared, it's saying that our model is doing a very bad job. So we haven't done a good job of kind of picking and optimizing which factors are good predictor variables and kind of help us kind of understand which cells are dying. So then we would go back to the drawing board and say, pick a few more, see which ones we have a good argument for, and then play around with it until we have something that looks good and something that works. But we don't have to do things that are sort of this complicated and this fancy. These are sort of powerful tools, but they're a bit more work. There are other things that we can do. So let's switch back to doing a default analysis. Analyze our data again. And let's look at some more kind of straightforward, simple tests. So in the first tutorial, we looked at sort of comparing between like a control group and an experimental group. But what if we want to compare within samples and like do a paired test? What we can do is a paired box plot, and we can choose sort of two variables to look at. Let's look at the, let's say, number of center cells versus the number of border cells. And you'll see in this time, instead of it being, showing you control versus experimental, it's showing you a pairwise comparison within the control group, sort of their center versus their border, and for the experimental group, which is labeled here at the top, ref2p, a number of center cells with a number of border cells. So for this particular data set, we have relatively small clones. We have a lot more border cells than we have center cells. And again, we can customize and export these plots. Something to note is that when you're doing these kind of tests, when you pick your graph type, the stats will automatically update. So for instance, in the previous tutorial, we would do between experiments like a student's t-test or a Wilcoxon-Mann-Whitney u-test or something like that. Whereas now, because these guys are paired, we do a paired t-test or a Wilcoxon signed rank. And we can also do a scatter plot, kind of look at two variables. Something random. And so we can look at sort of two different factors, and we can do sort of correlation tests as well to look at sort of how two variables look together. And again, this would all be put out in the statistics output tab. We can also look at individual clones. So to do that, you load in your clone tracking analysis files, and these operate exactly the same as your whole disk analysis files. Now let's say the only difference here is if we look at the analyzed data table, instead of it being an individual cell or a whole sample, it's actually just going to be each row is going to correspond to a individual clone. Um, and so each of these dots here is showing sort of the volume of an individual clone. And you see we have just a lot more measurements, so it gets kind of a bit overwhelmed there. So you might want to do a violin plot that might make it a bit easier for you to see what's going on with your samples. And that's sort of your breakdown of the sort of the features that are incorporated in the graphing and statistics component of the PECAn package.